We are now starting Chapter 8, which is about cell reproduction. This is entering into a new unit which is very different from the unit that we finished before. It will require a different mindset because in the last chapter we were able to physically see everything that we were talking about. We could see or imagine the living things and their interactions with the environment, but in this chapter we're going to have to rely on models, diagrams, and pictures, and even some animations to see or imagine what's going on inside the actual cell. We can't really see what's going on unless we were to use a microscope, and even then we can't see what's going on inside the nucleus, which is mainly where we're going to be focusing all of our studies in this chapter. So we're going to have to use our animations, we're going to have to look at videos, we're going to have to use models in order to imagine the small particles and the small structures that we're going to be looking at and try to envision what's going on. So this chapter is going to be a 3-part video series, and today we're just going to start talking about section 1, which is about chromosomes. So let's get started. Whoops. Okay, so we've all heard of DNA before. DNA stands for deoxyribonucleic acid, and this is the molecule that holds the blueprint for all of the inherited characteristics of every single organism that's living. But maybe you haven't heard of the term chromatin, and chromatin, in a very general sense, is the molecule of DNA that has these proteins called histones, and the DNA wraps itself around the histones. So DNA by itself is just the molecule that holds all the genetic information, but chromatin is DNA plus this protein that we call histones. So if we take a look here, we have our first stage, which is just DNA by itself. And remember, DNA is the molecule that holds all the genetic information. We're going to talk much more about DNA in our DNA section, where we're going to talk about all the nucleotides and molecules that make DNA and make up the structure. But just for now, we just have to know that DNA holds all of the genetic information for every single living organism, and DNA is found inside the nucleus of cells. Now DNA, like I said before, when it's wrapped around these proteins called histones, we call that chromatin. And the DNA doesn't change at all. It's just called chromatin when it has the proteins in it. When we have a cell that's ready to divide, we have to condense the chromatin. So chromatin, when it's, not in a non-dividing cell, it almost looks like a bowl of spaghetti, where the nucleus is the bowl and the chromatin is the spaghetti. It's this loose, free-flowing, elongated, spaghetti-like structure when you're looking at it from the outside of the nucleus. When cells are ready to divide, they have to condense this chromatin. And what happens is the chromatin winds up in itself very, very tightly, and it forms structures that almost look like X's. And we call those X's chromosomes. So just to recap, DNA is just the genetic information molecule by itself. Chromatin is DNA plus the protein called histones. This occurs in a non-dividing cell. Chromatin looks like spaghetti. And when a cell is ready to divide, the chromatin will wind up on itself and it'll coil very, very condensedly, and it will form these structures that look like X's, and we call them chromosomes. So here's a visual of what we were just talking about. So here is the DNA molecule all by itself. We have our double helix, so this is 1 helix, this is 2. These are the nucleotides here in the center. Like I said, we'll be talking about that much more in depth in our DNA chapter. So if we look, this is zooming in, we zoom out a little bit from the DNA molecule, we see that it's wrapping around these red structures, right? These red structures are the proteins called histones. So here we have our chromatin, and we're zooming out, And as I said before, when you zoom out and you look at chromatin from a farther angle, or a more zoomed out angle, it almost looks like spaghetti. And like I said, this is still found in the nucleus of the cell. And when the cell is ready to divide, it continues to condense and wind up on itself until it's tightly coiled into a structure that we see here that looks like an X, and we call that a chromosome. So let's look a little bit more closely at chromosomes. So a chromosome by itself consists of two identical halves, and we call them sister chromatids. So if we look here, what I don't mean half by half up here and half down here. That is incorrect. A chromatid is a vertical half. So this right here, that one piece, that is a chromatid. This piece right here, that is a chromatid. Together they are called sister chromatids, and they are combined or attached by this center piece right here in the center that we call a centromere. So that word kind of makes sense, right? We're talking about the point at which two chromatids, one chromatid, two chromatids, attach at the center, and that center point is called the centromere. So all species have DNA, and within their DNA code, they have specific genes that code for different characteristics. And this is what makes all organisms and all species different from each other. The fact that each species has a different number of chromosomes allows for a certain amount of genes to be produced, which allows for these specific characteristics to come out. So as you can see here, humans, we have 46 chromosomes per cell. This means per cell. Dogs have 78. Let's see, penicillin, which is a bacteria, they can have between 1 to 4 depending on the strand of penicillin you are looking at. Corn has 20. So just because a certain organism has a certain amount, let's just say like humans, we know that we are a very complex species. We have 46. Dogs are not as complex as us, but they have 78 chromosomes. So the number of chromosomes doesn't directly affect how complex the organism is, it just shows how many genes are expressed per cell. So there are two types of chromosomes that we need to talk about, and we have sex chromosomes and autosomes. So let's look a little bit deeper into that. Sex chromosomes are going to determine the gender of the organism that contains the chromosomes. So if the organism is a female, it will receive two X chromosomes. Now the sex chromosomes, there's only two of them that you can choose from. You can either have an X or you can have a Y, that's it. So if you're female, you are receiving two X chromosomes, and if the organism were to be male, it would receive one X and one Y chromosome. So every other chromosome that we have is not considered a sex chromosomes. They would be considered autosomes. So we have two sex chromosomes. They can either be X and X if you're female, X and Y if you're a male, and then all the other chromosomes that we have are considered autosomes. And they do not represent gender. They represent all the other possible genes that we have. So it could be hair color, eye color, height, personality, specific hereditary traits, absolutely anything. So the way that autosomes are arranged, every single chromosome has a pair, and they always hang together like a pair. And the reason why they find each other is because they're not identical copies, but they share the same, or they express the same genes. So in order to find the homologous pair, we call them homologous chromosomes or homologous pairs. And the reason why we call them that is because homo means same, right? So they are chromosomes of the same type, but they are not identical. They have similar shape, similar size, and they carry genes for the same traits, but that doesn't necessarily mean that the expression of the traits are the same, because you're getting one copy from your mother and one copy from your father. So, your mom could have green eyes and your dad could have brown eyes, but these two chromosomes are going to carry the gene for eye color, not necessarily being the same color. Oops. So the way that you're able to look at chromosomes and see if they are homologues is by looking at the pattern of the genes. So we have A and 4 being the same, because if you look at the banding here, okay, the shape of the banding, the pattern of the banding, the same size of the chromosome means that they carry the same traits. So B and 3 would also be homologues, as we call them. C and 1 would be another pair of homologous chromosomes, and D and 2 would be another pair of homologous chromosomes. And again, just from this activity, little activity here, you're really just looking at the pattern of the banding, but you would also, in a karyotype, you would look at the specific shape of them as well. So here we have an example of a karyotype, which is essentially a map of all of your chromosomes. If you were to get karyotype testing done, this happens a lot, fetal karyotype testing when women are pregnant, they look to make sure that they have the right amount of chromosomes, they all have their pairs, and they also look at gender that way too. So remember I said that all humans have 46 number of chromosomes, but we don't really go by the actual individual number, we look at their pairs. So essentially we would have 23 pairs of chromosomes, right? If you take 46 divided by 2, right, you're going to get 23. And if you look here, they are numbered in order. They go from largest to smallest, and each pair carries the same set of genes. But remember, one chromosome is from mom, one chromosome is from dad. So the actual expression of the gene can be different. So these two, right, similar size, similar shape, they get paired up together. Same thing here. They go from largest to smallest. And then our last pair here, which isn't numbered, remember I said we have 22 pairs of autosomes. Autosomes are the non-sex chromosomes, and we have one pair of sex chromosomes, which gives us that number of 23. Here, obviously, this would be a male, right, because we have an XY here, so that would be this would be a karyotype for a male, and this is a healthy male karyotype because every single number here has its own pair, and the pair matches up, and they have the same shape and the same size, and nothing's missing and nothing's duplicated or added. So this would be a healthy male karyotype. As stated before, we have all humans have 22 pairs of autosomes and one pair of sex chromosomes. So that gives us a total of 23 pairs of chromosomes, but if you were to look at that individually, every single chromosome individually, humans have 46 chromosomes per cell in their body. If you were to not have 46 chromosomes, you would then either have some type of genetic disorder where maybe you have an added chromosome or you have a deleted chromosome. And we'll talk about that much more in our genetics section when we talk about genetic disorders. So of course, not only, to make this a little bit more complicated, not only do we have to realize that we have two types of chromosomes, which would be our sex chromosomes and our autosomes, but within those cells we have two different types of cells. So within each we have two, okay, let's start about the types of cells. So we have body cells first, which we can also call somatic cells, and then we have sex cells, which are called gametes. Somatic cells, or body cells, they have a complete number of chromosomes. So that's the complete 46 individual, the complete 23 pair. They have one pair of sex chromosomes. Please don't get sex chromosomes and sex cells confused, okay? Sex chromosomes are just the gender identification, 1X, 1Y, or 2X's. But a body cell has them too. They have the 22 autosomes, 22 pairs of autosomes, and the 1 pair of sex chromosomes. The sex cells, or the gametes, don't have the homologous pairs. They just have one of each of all of them. So they don't have 22 pairs of autosomes. They just have 22 autosomes. And they have one sex chromosome. So it's either 1X or 1Y. These are the cells that help fertilize the egg. So the egg for the female is going to be the gamete, and the sperm for the male is going to be the gamete, so that when they come together, they will then form enough genetic information for the new generation for the body cells. So let's take a look at some terms here that can help us describe the differences between the gametes and the body cells. So we have this term here called diploid, and diploid is referred to as 2N as shorthand. And all diploid means is that you contain 2 autosomes per pair in 2 sex chromosomes. So this is what we were talking about before, where in each diploid human cell, remember we're just talking about humans, every organism has their own number, but in each diploid human body cell or somatic cell, there are 46 individual chromosomes. Or you could say there are 23 pairs of chromosomes, 22 of them being autosomes, 1 of them being sex chromosomes. These are the cells in your body. These are every other cell in your body except for egg and sperm. So this could be skin cells, brain cells, heart cells, liver cells, stomach cells, any cell, all right, any cell in your body. If you are human, they are diploid. There are 46 chromosomes or 23 pairs. The haploid cells, or you can think of half, H-A-L-F, haploid, right? It's half the number of the diploid. And the haploid cells, or the gametes, right? These are sex cells. They are only going to be egg or sperm. So if you are a female, your haploid cells are, or your sex cells are called eggs. If you are a male, your haploid cells or your sex cells, that is your sperm. And when you reproduce and the egg and the sperm meet, right, if you double the number of 23, you're going to get 46. So that is how the next generation is able to have the same amount of body, or sorry, not body cells, but same amount of genetic information in the body cell. And the way that the gender is predicted or created is the egg is always going to offer the X, but is the sperm going to offer a Y or an X, because males are XY. So that's how it's 50-50% for the gender for the next generation. So far we've just been talking about haploid numbers and diploid numbers for humans, right? The haploid number for humans is always going to be 23, and the diploid number for humans is always going to be 46. But those numbers are really easy to memorize, but really when you look at it, all you're doing is taking the diploid number and dividing it by 2 to find the haploid, or if you have the haploid, you're going to multiply it by 2 to find the diploid, because haploid is just half of the diploid number. So we could find any haploid number and a diploid number for any organism. So let's take a look at this chart right here. If we were given the haploid number, right, which just represents N, for an amoeba is a unicellular organism that's normally found in ponds, okay, the haploid number is 25. So if we were to find the diploid number, all you'd have to do is multiply it by 2, and now you have 50. Okay, same thing for the chimpanzee. If the haploid number is 24, then the diploid number is 48. Earthworm 18, 36, right? So we're multiplying by 2 if we're going from haploid to diploid. If it's the opposite, so let's just say for a fern, this is a type of plant, if the diploid number of a fern is 1010, we would divide that and we would get 505. Remember, if we're going from haploid to diploid, we are multiplying. So for the hamster, it's 22. Diploid would be 44. A honeybee's diploid number is 56, 28. Okay, you try it on your own. Humans, well, we've already memorized this one, right? 46, 23. And an onion, 16, it would be 8. Okay?